joining our webinar today. So today I'm going to be talking to you about sample preparation for single cell analysis, basics and beyond. My name is Catherine King. I'm a field application scientist with 10x Genomics based in Melbourne in Australia. So to get started, I just actually wanted to give a bit of an introduction to the 10x Genomics team in Australia and New Zealand. We all relatively, we've got a new team here, so I wanted to really take this opportunity to introduce our team. As I mentioned, my name is Catherine King. We also have two science and technology advisors, that is Yvonne and Jeff, and we have our 10x business manager across ANZ, Brett Kennedy. I can also introduce to you our the Millennium Science team, who I'm sure that many of you have worked with already. So we work very closely with Millennium Science. Millennium Science is our official distributor here in the ANZ region. So that we have Daniel Dugalensky, who is the 10x business lead, and Paul Gooding, who is the field application scientist for Millennium Science. So Paul and I work really closely together to support customers across the region. So before I really kind of start talking about sample preparation, I want to give a very brief introduction. I'm not going to spend too long here on just what you can do with the single cell solutions from 10x Genomics. So we have multiple solutions that you can use for your studies. So you can combine surface protein expression with gene expression analysis using our feature barcoding technology. If you are interested in immune profiling, you can use our immune profiling solution in combination with gene expression, cell surface protein, antigen specificity, and look at full-length TCR and/or BCR sequences. If you are studying cell differentiation or lineage mapping, you might be interested in our epigenomic assays. So we have our ATAC assay, or we have our combined multiome ATAC and gene expression solution, where you get both of those modularities from the same single cell. Finally, if you are interested in doing CRISPR screening and looking at CRISPR, you can actually look at permutation profiles of individual single cells using our feature barcoding technology. So just kind of thinking about sample preparation here as an introduction. So depending on what you're measuring, you're going to have different inputs into these various assays. So if you're looking to assay cell surface proteins and RNA, then you're going to have to use whole cells as an input, because we need that cell membrane, for example. If you are looking just to look at gene expression and your RNA content in your cell, then you really have the choice between using whole cells or nuclei as an input, and that decision we'll talk about more today in this presentation. And if you're looking to do any of our epigenomic assays, where you're looking at the open chromatin and chromatin structure, then nuclei is absolutely your required input. So just really introducing here that the input into our assays is going to be highly dependent on what you're trying to achieve. So how does our technology actually work? Well, let's first just again mention our inputs into the assays. So that could be single nuclei or single cell suspensions. It could be single cells that transduced with a barcoded CRISPR guide RNA, labelled with a barcoded antibody, or labelled with barcode peptide MHC multimers. These are kind of the inputs that can go into our assay. They get loaded onto the chromium chip, where they then mix with our 10x barcoded gel beads. The gel beads are anchored with unique 10x barcodes, and these are what allow us to distinguish between individual cells when we form our gems or gel beads in emulsions. So this technology was really carefully designed to allow us to get the highest throughput and cell recovery, as well as low cell multiplets. So with that very brief introduction, I'm now going to really take a deep dive into single cell sample preparation. So at 10x Genomics, we really define sample preparation in 3 ways. It is what you bring to the experiment. It's a workflow and a set of decisions. And it's I guess an understanding that determination of quality is absolutely critical. So we need a really high quality sample, and we need to have a way of determining that. So the input into the single cell assays are single cells or single nuclei suspensions, whether they have labeling or with antibodies or so forth, but they are single cell or single nuclei suspensions of the highest possible quality. To achieve a high quality cell or nuclei suspension, it's important to first consider what you're bringing to the experiment. So, what organism are the samples from? Are you familiar with working with these organisms to isolate your samples? What specific tissue or cell type are being used? Are you aware of complexities with working with that particular tissue or cell type that is going to make it difficult or so forth to generate a single cell or nuclei suspension? So, for example, are you aware that this is a particularly hard to dissociate tissue, or there is a lot of debris expected in this sample? And how are you going to generate these single cell or nuclei suspensions? Have you already got a protocol that you're using in your lab? Or are you starting from scratch and you're needing to, get out there and figure out what protocols there are that are going to be suitable for single cell assays? And I think the goal of really asking yourself these questions is evaluating where your starting point is and the gaps in your knowledge that you need to really basically fill before you can start to prepare your sample in a sufficient way for the 10x assay. So here we're looking at the kind of key workflow steps in sample preparation. We have kind of sample collection, sample preservation and transport if required. We have to then take that sample and generate a single cell or nuclei suspension. This may involve a tissue dissociation step. We may need to enrich or deplete a certain population of our cells. For example, we might be looking to enrich a certain cell type, or we might need to remove dead cells because our viability is low. And then we do need at the end to do a sample QC. So that's kind of a typical sample preparation workflow. The steps in blue are really critical steps and required steps. The steps in grey are potential required steps or optional steps. And what I will first highlight is that we have many demonstrator protocols and a Q&A section that is available on the Genomics Support page, which do have kind of guidelines to help you with each of these steps in the sample preparation workflow. So the Genomics Support page is at support.10xgenomics.com. If you come to this page, you will see various assays listed. And if you select any of these assays, you'll see the sample preparation tab. Now, under this tab, you will find some general sample preparation guidelines, such as, our general cell preparation guide, which we'll talk about in a moment. We have guidelines around accurate cell counts and so forth. We have demonstrated protocols for preparation of very specific sample types, so that you may be using one of these sample types, or you may use this to help design your own sample preparation workflow. And we have some technical notes and demonstrated protocols around sample improvement. So including protocols for enrichment of T cells from dissociated tissues and removal of dead cells, for example. So the aim of all these, we're always generating new documentation and new demonstrated protocols, and they will be added as we develop them. And this is the work that we're doing in-house, trying different samples, getting protocols that are working well in-house. And the idea of these protocols isn't to take them and use them as is. It's there to use as guidelines to optimize your own sample preparation to use in that way. We also have that Q&A section that can be found on the Genomics Support page at the very top. If you click here, we have a really great resource for each assay in regards to sample preparation, and with some answers to kind of detailed answers to some very commonly asked questions when it comes to sample preparation. So I mentioned our cell preparation guide. This is a really great place to start if you're really new to single cell assays, because it really gives an overview, a very brief overview and general kind of protocols for key things that were required for a good 10x Genomics Single Cell sample preparation. So this includes washing and resuspending your cells, straining, counting, determining your cell concentration, and working with limited cell suspensions. The key thing, and what I really want to emphasize, is that quality is absolutely critical. So your input into our assays is very important to get high quality data. And so by having high quality samples, what we mean is having nice, highly viable cells, if you're working with cells, and this should be a viability of at least 70% or greater. And in fact, we want much higher than this. You want to be aiming, if possible, 90% and above. Now, obviously, cell viability is not applicable when we're working with nuclei. Nuclei will all stain as dead. However, we do want a nice, healthy population. So, sorry. And so I'm just going to take a drink. Sorry about this. So when we're working with a single cell suspension, we need it to be very healthy. And so what we mean by healthy is in a minimal state of biological decomposition. So viable cells are going to have lower rates of RNA leakage and degradation than apoptotic or dying cells. If we have RNA leakage, we're going to have background, and this is going to be background in our data. If we have RNA degradation, that basically means loss of signal. And we're not really going to be looking at the true transcriptome that we're really aiming to see. We want our cells or nuclei to be physically intact, so in a minimal state of physical decomposition. Again, we know that if our cells or nuclei are physically breaking down, we're going to have that RNA leakage and degradation. We also need a suspension that is clean and free of aggregates and clumps, subcellular debris, and free-floating RNA. So the goal here is to maximize your data output by minimizing dead cells and background signal. So our general recommendations in order to have this nice, happy, healthy, intact population is to minimize cell lysis and loss. So we want to treat our cells, if we're working with cells, but also nuclei, with really gentle pipetting using wide bore tips, because we know if we handle cells too roughly, many will lyse, releasing this background RNA into the cell suspension buffer. We want to always, when we're starting our sample prep, we are going to be taking them from a place where they're happy. So if we're taking our tissue, we're taking it and we're dissociating those cells, so we're taking them from a place where they're happy, and they're going to be on a decay time, like on a decay from that moment till you get them loaded onto the chip. So you want to work quickly, minimizing unnecessary handling steps and minimizing the time between sample preparation and loading onto that chip. We recommend counting your cells regularly. By assessing the viability and the amount of debris and aggregates throughout the workflow, this allows you to basically make critical cell preparation decisions as you go. If you can see that you still have a lot of debris, then you know that you need to do some more filtering or cleanup steps. It also allows you to assess where you are with your cell number and understand how much have you got to work with, and estimate that final resuspension volume, which is critical. So now let's look back at our sample preparation workflow and take a look at some of these key steps and some of the considerations that you can have here. So first of all, let's look at sample types. So the best input into a single cell assay is fresh cells or tissue. If that's not possible, of course it's not always, then your next best option is cryopreserved cells. All of these as inputs into our solutions allows you to look at not only gene expression, but you can also use the various extensions of our assays, such as doing CRISPR screening, cell multiplexing, looking at cell surface proteins using feature barcoding, as well as our VDJ solutions and immune profiling as well. Now, in some cases, if you were, for example, using our epigenomics assay, then nuclei is your required input. You may also have other reasons to use nuclei, which we'll look at in a moment. And in those cases, the only kind of, well, not the only, but the assays that are available to you are our gene expression assay and cell multiplexing. If you are working with SNAP frozen tissue, then your only option is going to be to isolate nuclei. And this is because when you've SNAP frozen tissue, you've essentially, the cell membranes of the cells in that tissue are going to be compromised. So we know that your nuclei is really your only option. And with that, they're a little bit more unstable. So really you are only going to be able to look at using gene expression, and those feature barcoding, for example, is a lot more challenging there. And this, working with SNAP frozen tissue, isolating nuclei is technically challenging, and it will require optimization in your hands. We do have a fixation protocol for working with a single cell suspension. This is our methanol fixation demonstrated protocol. This is very technically challenging as well. So it does have limited capabilities with these cells, and we do prefer, if you're looking to preserve your cells in order to process later, then in these cases we would always suggest cryopreservation of your cells instead, if possible. On this note, and I will come back to it at the end of the presentation, but we do shortly, in the near future, are going to be releasing a fixed RNA profiling solution. This is going to allow you to fix your cells for single cell assays, allowing some of our feature barcoding and assay extensions, such as cell surface protein analysis. So watch this space for that one. So we know that many people work with tissues, and this slide says from clinical samples, but really you could be collecting tissues from patients or from mice, or however, where that tissue is from. And I think one really crucial thing to be aware of is that timing is absolutely critical. So we talked about earlier how once you have taken your sample from their happy, environment, that they're going to be on a path of kind of decay, and their quality is only going to start reducing. So you want to minimize the time of taking that tissue that you want to work with and to either preservation or sample preparation for the single cell solution. You want to minimize the time that it's left outside those optimal conditions. So a lot of people ask us, well, how do I preserve, for example, my tissue if I want to work with tissue, but I can't prepare it straight away? So there are options out there. These are just some options for you to consider. They are going to be highly variable depending on the tissue type you're working in. All of these would require optimization in your hands, but there's some things to consider. So if you are happy to work with nuclei, and that is going to be applicable for the assays that you want to run and the applications that you're trying to perform, then snap phrasing of that tissue might be a good option. If, however, you want to work with single cells, you have reasons that you want to perhaps enrich for specific cells or do feature barcoding, then you can consider there's options such as tissue storage solutions and cryopreservation in media. This will not allow long-term storage of those tissues. Actually, it's kind of a maximum of 72-hour kind of short-term storage, but this might be an option if you cannot isolate your single cells immediately, but you do want to work with single cell suspensions. Once you can isolate those single cells, then you can go into our solution or potentially cryopreserve those single cells as well. So now let's talk about actually generating a single cell or nuclei suspension. So first of all, you're going to have to think about your starting sample. What are you working with? So if you're starting with cells, then of course are they primary or cultured cell line? And if it's a cultured cell line and it's adherent, then you're going to have to do an enzymatic dissociation in order to get your cells into a single cell suspension for the 10x solution. Now, if you're working with tissue, you have, I guess, a few more options and considerations here. So As we spoke about earlier, if you have frozen tissue, then your only option is going to be a mechanical disaggregation to isolate nuclei. Those membranes of the cells in that frozen tissue are going to be compromised, which means this is really your only path and will require optimization. Now, if you're working with fresh tissue, you have some options. So if you want to work with those single cells because you want to potentially enrich for a specific sample cell type, you want to look at cell surface proteins, or perhaps you have a method in place to dissociate those single cells and that's working well, then you'll use an enzymatic dissociation into a single cell suspension. There may, however, be times when you want to take your fresh tissue and it's more challenging to create a single cell suspension. There might be reasons why you don't want single cells, for example, their cell size, which isn't compatible with our solution. And in these cases, you might choose to work with a quicker kind of mechanical disaggregation process into nuclei suspension. So where can you go to for resources? If you want to start working with tissues, where can you look for resources around tissue dissociation for your particular tissue type? And so we have multiple places we would recommend to look. So we have our demonstrated protocols on the Genomics Support site that I've already mentioned. So we have various dissociation protocols for neural tissue, for tumors, demonstrated with various tumor types, and also details around freezing and thawing some dissociated tumor cells on our website. We have a publication database on the 10x Genomics webpage as well. This is a really great place to find publications using our technology, and you can search by species and tissue types to see what others in the literature are using to dissociate their tissues. We have various compatible products. So we have, actually it's in some of our demonstrated protocols, we use the Miltenyi solutions. So that includes the Gentle Max dissociator and dissociation kits. So you can see that used in our tumor dissociation demonstrated protocol, and they are a great resource, Miltenyi, to get more ideas and around dissociation of your tissue. We have also a section on the 10x Genomics support page for, oh, sorry, not the support page, just 10x Genomics website, where you can find customer developed protocols. So these are not generated or ultimately supported, I guess, by 10x Genomics, but they're a great resource, and we know that many people are using these protocols successfully for their 10x Genomics assays. So that's a great place to review what's there as well. And finally, I always recommend that when people are starting with a new tissue type, to check out the Worthington Tissue Dissociation Guide. So this will give you ideas on how to dissociate your tissue and link you to cited publications where you can get details about how they perform those dissociations. So this is just an example of our tumor dissociation for single cell RNA sequencing demonstrated protocol that's available on our 10x Genomics support site. It was demonstrated on mouse colon, breast, and melanoma samples, and this utilizes that Miltenyi tumor dissociation kit and the automated Gentle Max dissociator. We also have guidelines in here in the appendix for improving cell viability as well. So that's a great resource. So we'll switch gears a little bit and talk about why you might want, or some instances where you might want to consider nuclei as a sample input into our 10x Genomics single cell assays. So if you're using any of our epigenetic assays, so whether this is our standalone ATAC assay or our multiome combined gene expression and ATAC assay, then nuclei are your required sample input. Now, if you are working with an incompatible sample type, you might choose to also use nuclei. So example is that SNAP frozen tissue we mentioned earlier, where we know the cytosolic, the cell membrane, this cytosolic RNA is compromised. There might be challenges that are inherent to the specific tissue or cell that you're working with that makes it easier just to isolate nuclei. So it might be difficult dissociating that tissue, or it might be the cell size is an odd shape or too large. The microfluidic channels, we support up to 30 microns. So you might choose to therefore use nuclei instead. You also might choose nuclei if you're working with a sample of unknown quality. So not kind of low quality, but more it might be the cell integrity may be damaged, such as in preserved, archived samples. Then you may choose to isolate nuclei in these examples too. So although there are many variations of nuclei isolation protocols, the key steps remain the same. So the idea is that first you're going to take your sample and incubate it in a lysis buffer. So this lysis buffer is going to depend on the assay that you're looking to run. And so you need to make sure you're working with a protocol that is compatible for the assay that you're trying to run. So to explain that a little further, so with our epigenetic assays, our ATAC or multiome solutions, we include a digitonin in that lysis buffer. Digitonin will actually poke holes and permeabilize the nuclear membrane, which is required for the transposition enzyme to enter. Now, that is not something we want to add if we're not doing ATAC with our nuclei. So this is how the lysis buffers will change and be different for the different assays that you're trying to run. Now, once you've lysed and you've released your nuclei, you're going to try to break up your cells and tissue by either using various pipetting techniques or with a pestle in the case of tissue, and then you're going to release and lyse and release your nuclei. From here, you're going to basically take that sample through a filter in order to remove large pieces of debris and break up clumps. You're going to then isolate and spin down and wash your nuclei in order to remove ambient nucleic acids and also debris, before then resuspending them in the appropriate buffer. And again, resuspension buffer is going to differ between the type of assay you're running. So that will differ for gene expression assay versus, say, ATAC or multiome assay. So you need to make sure, again, that you're following the appropriate protocols. So although the steps are very similar here, there's definitely going to be optimization required of these steps for your sample. So you do need to optimize your lysis conditions so that you are getting high quality nuclei and not over lysing. If you're working with particularly samples with large amounts of debris, you're going to have to consider ways to make sure that you can remove that debris. And that may be through sucrose gradients, it may be through flow sorting as well. So how do you optimize your own nuclei isolation? So we have various recommendations. So lysis buffer. In our protocols, any of our nuclei isolation protocols, you'll find that we generally either use a 1x or 0.1x lysis buffer. Now, you want to use, we generally, for our protocols, if the protocol is for cells that are already in suspension and they don't require dissociation, we use a stronger lysis buffer. For those that are from tissues, we start with a more gentler approach. So that is something, this is a great place to start, but you might find that you need to reduce the stringency of your lysis buffer if you're finding the lysis you're getting over lysis and it's not performing well. So how do we know if our lysis is performing well? So we want to do a lysis timeline for each sample type. So you take that lysis buffer, you're incubating it with your sample for a certain amount of time, and you'll use a broad kind of timeline if you're working with quite tough cells. If you're working with very sensitive cells, then you might be looking at, just a minute or two between your timeline steps. You're going to be taking them during those lysis times and checking the nuclear membrane, checking the viability count in order to see what their best lysis time is. And we're going to show that on the next slide. Other things you can optimize with lysis, with nuclei isolation, is recovery of your nuclei. So if you find you're not recovering enough nuclei, then you can increase your spin time. And we'd always suggest using a swinging brackets rotor for your centrifuge when working with nuclei in order to maintain and not lose too many. In terms of once you've got your nuclei, you're going to resuspend them in the resuspension buffer for the particular assay you're working with. This is a really key QC step. So if you resuspend your nuclei and you're seeing just a few clumps and debris, that might be normal, and you can filter using a FLOMI filter, assess and continue. Now, if you're seeing major clumps of your nuclei when you're going to resuspend them, that is a big red flag that potentially those nuclei membranes are too compromised. It's likely meant that the lysis buffer or time was too harsh or too long, and you need to go back and optimize that lysis condition. Finally, we want to do count and QC. So you're going to use your counting to visualize your nuclei and make sure that they're clean and not clumping. For counting, nuclei can be challenging to count, so we recommend, where possible, using a fluorescent dye for increased accuracy. So this might be like AOPI. And this is where, and QC is where we can check the nuclei and make sure that we're happy with their quality. And we would do this by using a viability dye, checking for the live cells, and also assessing the nuclear membrane. So let's have a look at that in more detail. So what do we expect to see if we have optimal lysis? So what we want to see is less than 5% live cells, but ideally maybe a few live cells still remaining. If we have 100% dead, it can mean that we've actually gone too far and we've overlyzed the sample. So if we're doing our lysis time point, we want to look at the point where we're just getting less than 5% still remaining, but perhaps a few in there. And we want to visualize those nuclei and they're clean, clump-free. We know we have suboptimal lysis conditions. If we have greater than 5% of live cells still remaining, we haven't lysed them, or conversely, we've overlysed and we have lots of clumps and debris. This gives us a yes/no answer. Have we lysed enough or not? But it doesn't really tell you the quality of your nuclei. So we also always recommend assessing the nuclear membrane quality under a higher magnification microscope. So this would be, this is available on our website as well, these images, but what we want is nuclei that look like those in A, ideally, or B, where we get a nice smooth nuclear membrane around the outside. What we do not want to see is what we can see in C and D, where you can see this membrane is compromised. We're kind of getting extensions of that membrane. It's opening up, and this is what we call blebbing, and this is what we would need to readdress our lysis conditions if we're seeing nuclei that look like this. If we ran this with any of our solutions, we're likely to end up with lots of ambient RNA in our data, and ultimately it's not going to be what we're looking for. So definitely want to optimize those conditions. Okay, so now let's talk about sample enrichment or depletion. So why would we, what ways can we clean up our sample and enrich our population? So washing our population will allow us to remove ambient RNA and some dead cells. If we filter, we're able to remove large debris from our sample, and we may also choose to enrich or deplete for a specific population. So we might be depleting dead cells, and we might be enriching, for example, a specific population of T cells, if that's of interest. So these are the kind of sample cleanup and enrichment techniques we can use. It's always important to do washing and filtering to make sure that we have a really clean sample, but of course enrichment depletion is going to be highly dependent on the sample that you're working with. So getting a combination that works for your sample preparation method and your sample is going to be something that you need to optimize. You want to consider how many cells you have, how sensitive they are, how much time you've been working with them, and if they're on a rapid decay path. So it's really a balancing act at what's the minimum amount of minimal amount of sample manipulation that will deliver the sample quality and the input into get the application metrics that you want. So here's a table that just lists some common methods for sample cleanup and enrichment. So this by no means is all the methods that you could use. It's just some examples of some of the methods that we have used in-house in our DNS demonstrated protocols. And you'll see all the methods across the top, and on the second row, you'll see in which method or which demonstrated protocol these methods were used in our protocols. So we have, we're going to be talking through a couple of these in the coming slides, but just kind of to quickly mention, like a thorough centrifugation technique, which is in the first column there, that's used in our PBMC protocol. This is a really thorough wash centrifugation method. It's really great if you've got abundant cells and you want a really nice, clean single cell suspension, and you'll get really clean data. However, it can potentially be too harsh, especially if you don't have enough cells, you lose too many cells in this, that it won't be applicable for your sample. On the other hand, you've got a gentle centrifugation technique. This is demonstrated in our thawing-dissociated tumor cells. So the washing is not as stringent. You ultimately, it won't clean the sample as well. So you may see higher background in your data set, but it's a great option if maybe you're working with limited numbers of cells, you need it to be gentle, you might be working with more sensitive cells. So this is where, I guess, highlighting these two is highlighting where you need to consider the sample you're working with and what can you do to clean up that sample as best as possible without compromising that sample quality further. So we list here as well magnetic bead depletion enrichments and density gradients, which is great when you have abundant cells or nuclei for cleanup, and FACS enrichment. So we'll talk about the magnetic steps and in FACS enrichment further here. So separation method of dead cell removal. So you can use various kits to remove dead cells. So for example, the Miltenyi kits, and this is we have a demonstrated protocol around this. So you're basically able to bind magnetic beads to a surface protein expressed by apoptotic cells. The beads will bind to those dead cells and you're able to collect them through a column so that you're only obtaining live cells at the end. So this is great when you're working with abundant sample, but it's got poor cell viability, and it's a quick way to end up with a nice high viability population. Magnetic bead enrichment. So this is where you, instead of trying to get rid of the cells, you're obviously trying to enrich for them, and you can again use a magnetic bead process for this. And again, companies such as Miltenyi offer various options here. So this is great if you want to enrich for a certain cell type, you don't have maybe access to flow cytometry or that expertise, or that's going to be far more challenging than performing the magnetic bead enrichment step. So this is a great option for really enriching for a cell type of interest. What you do need to be aware of with this technique, however, is that you can really only isolate that specific cell type, but it's not going to be able to, once you've performed this, you can't then go and remove dead cells using that magnetic dead cell removal. So you need to be kind of working with a high viability sample so that you can move forward following this protocol. So another really great versatile technique, which we have a lot of customers using, is flow sorting. So this is where you could use antibody labeling or fluorescent dyes to select for cell types of interest. So it may be that you've just got a really a sample with a lot of debris and you're not trying to select for a specific cell type, but maybe you just want to clean it up, and we have lots of customers do that. So they select just for live cells or single nuclei. So this can be a really versatile, great technique, but you do need to make sure that you're working with a flow facility or personnel that have experience with flow sorting and that can, take into consideration ways to minimize loss of your sample and minimize the harshness on your cells. So we have some guidelines on our in the Q&A section for the sample preparation on the 10X support page for considerations for flow sorting of your cells. So we always recommend doing some washing and filtering of your cells for our single cell solution. So washing will allow us to remove ambient RNA and small debris. We ideally want to do 2 washes if that's possible based on your sample number and the stability of your cells. You're going to have to optimize the spin speeds and the times of those speeds to make sure you maintain your cell population. And we recommend using PBS with 0.4% BSE for our washes, but of course this can be substituted with various common culture buffers. And we have, again, on that Q&A section of our support page, a list of other cell culture buffers that we've used successfully with the 10X solutions. We want to remove large debris in cell clumps using a filter. And we have 2 recommended filters that we use in-house, and that's the Flomi cell strainer from Bellart and the Max Smart strainer from Miltenyi. Now, the choice between these really comes down to the volume you've got to work with and whether, ultimately volume and what is available. So the Flomis are great for a quick filter once you have your suspension ready for final QC because it just really attaches to your pipette tip and you can pipette through very quickly in order to have a final kind of filtration of the sample. So it's important to filter away aggregates and large debris because even if we have a sample that's quite clean from ambient RNA, if there is kind of any aggregates there, even a small number, if those microfluidics channels in the 10X chip can clog, they're 50 micron approximately in size. So if you have some aggregate and they clog, that can mean the end of that sample. So it's important to make sure that you have a nice clean single cell suspension or nuclei suspension. Okay, let's finally talk about sample QC a little bit. And when I talk about QC in this, I'm talking really about counting. So counting is incredibly important for the 10X single cell assays. And the reason for this is, we want to target a certain number of cells in order to get the data that you're interested in. If you count and it's inaccurate, then you end up loading too many cells. What that means is that you can be overloading the chip, you can end up with large numbers of multiplets, and you also need to then spend a lot more money on, say, sequencing to get the full complexity of that sample. Conversely, if you count and you overestimate the concentration, you end up loading too few cells, then you're doing your experiment, you're hoping you need a certain number for your data analysis and you're not going to end up with that yield. And it will be a very expensive experiment for not getting the cell numbers that you need for your analysis. So counting is incredibly important. So we recommend that when you're working with it, whatever counting method you use, whether that be hemocytometer or a countess or another equivalent automated counter, that you become very familiar with that counter, and that if you're using it for the first time with a certain sample type, make sure that you're testing that and making sure you're confident with those cell counts. When you're taking a sample to cell that count before loading onto the 10X chip, we recommend taking 2 aliquots from that sample and doing at least 2 counts from that from those aliquots, so that we're confident in the cell counts going onto the chip. Cell counting also has the added benefit, as mentioned earlier, that you're able to visually check your final cell suspension. So if you're checking and you see there's still debris or large cell aggregates, or if you're working with nuclei, for example, you see lots of clumping, this is going to be a red flag and it's going to suggest that you might need to do more with that sample and it might not be worth loading at this point. When you count, you want to make sure that you're working in the correct cell concentration range for your particular counter to get accurate cell counts. And when we have our final resuspension, we want to make sure we're working in that recommended concentration range between 700 and 1200 cells per microliter, ideally, for loading onto the 10x chip. So we always take that final count of our very final resuspension. So you don't count and then resuspend in the volume determined. You want to count your very final cell suspension. So there's several factors that influence the cell recovery from any of our assays. So this is going to be much of what we've talked about today. So the sample type and your upstream handling, the cell viability, the counting accuracy, the time that cells or nuclei are left on ice before loading onto the chip. So if we're spending too long, that can obviously increase cell clumping, decrease cell viability, which can influence how many we get, the number of cells that we get out when we run our assay. And then other kind of considerations here is just making sure we're always working with good pipettes. We're using accurate techniques in order to maintain the cell recovery that we're looking for. So with that, I'll just mention where there's other sample preparation resources available to you. So we have our demonstrated and protocols and technical notes, which I've already mentioned, that are on our support page. We have those to customer developed protocols and publications. We also have a series of 10Expert webinars that can be found on the 10X Genomics website as well. We have various seminars and webinars based on specific sample types and considerations. So for example, tumor dissociations as shown here. And we also have our support team, and you have access to both Paul and myself as your local field application scientists here in the ANZ region. Before I just finish up, I want to just mention some of the new products we have coming from 10X Genomics very soon. So, or actually maybe also what is just been released now. So we have our CellPlex multiplexing solution. We've got our new low throughput kit, and we've got our Chromium X and IX and high throughput kits that have all just launched. As I mentioned earlier, in the single cell space, we have a new fixed RNA profiling kit, which is really exciting for lots of people. It's going to allow you to preserve your sample for the single cell assays, but still allowing you to be able to do things like feature barcoding, which will be a huge advantage. We have 5' CRISPR coming, and we also have Beam or barcode enabled antigen mapping with our immune profiling solution coming soon. From our spatial platforms, we've got various advances coming in our Visium solutions, such as larger capture areas, a way to multiplex with protein on our tissue sections. We've got a CytAssist instrument coming, which will help with placing your sections on the Visium slide. And we've got a new high resolution Visium solution coming out too. So if you're interested in any of these products, please get in touch, because of course, we'd love to chat to you about them. And finally, I keep moving on, but I also want to mention that we have recently started some new meetings, which are called Meet with a 10Expert. It means that if you have any questions around your 10X assay, whether that be designing the experiment, or you're looking to help with grant applications or ideas, you want to understand the workflow further, or you're having questions around sample prep, then please reach out. And I believe Karen will be sending a survey at the end of this presentation where you can get in touch and ask for a one-to-one meeting with our 10X team here at the ANZ region to discuss your project.